Welcome to our first session after lunch, the Oak Restoration and Conservation session. Our first speaker is Lucy Kerhoulas. Lucy grew up in Northern California and is currently an associate professor in forestry at Cal Poly Humboldt. Her research focuses on tree and forest physiology, and her studies include redwood forest canopy ecology, conifer encroachment in oak woodlands, forest demographics, and tree drought responses. When not working, Lucy enjoys hanging out with her family and apparently climbing trees, as I noticed she was in the recent issue of the Save the Redwoods League magazine. Welcome, Lucy.
Hello, welcome back from lunch. I hope you had a good outing. And yeah, I'm really happy to be here talking to you guys about the influences of conifer encroachment and removal on ecosystem physiology and biodiversity using an Oregon white oak woodland in Humboldt County as kind of a case study. And before I get rolling, I just wanted to give thanks to, it definitely takes a village to do science, so I don't work in a silo. Thanks to our funders, all of my collaborators, the student assistants who helped with this work, and Humboldt Redwood Company for letting us do this research on their property. So I am lucky enough to be kicking off the session on oaks. And just a little background, these are these sort of iconic landscapes in California, and they're really important culturally to many indigenous tribes, and they're also biodiversity hotspots. So they're cool landscapes, but in recent years, they've been under a lot of different threats. So things like habitat loss due to urban and agricultural development, and the introduction of non-native pests and pathogens, and also conifer encroachment in the absence of fire. So in Northern California alone, in the last 50 to 100 years, roughly 30% of Northern California woodlands have been lost due to conifer encroachment. And we see regional widespread decline in oak woodlands across much of the Pacific Northwest due to this. Oaks are a shade-intolerant group of plants. And so when they get shaded out by the conifers, they don't do well. And so we're seeing a loss of oak woodlands. Here you can see this was Maddie Shriver's master's thesis. This figure comes from that part of our group, our collaborator group at Cal Poly Humboldt. And you can see they pretty compellingly kind of documented the black portions of the columns and the white are oak regeneration. And then you can see starting in the 1850s, this decline in oak regeneration, and then the rise of the Doug fir establishment in red. And so our crew at Cal Poly Humboldt decided to do an investigation using an oak woodland near the university. And we had a lot of collaborators, and we looked at a lot of different questions, but 15 minutes is short. There's so much to say, so little time. So I'm just going to talk to you today about two questions that we investigated as a team. And I'll say this was a master's thesis, Gabe Goff's master's thesis, and it was just published in Restoration Ecology. So if you want more details, you can check it out there. So the two questions that I'm going to talk with you about today are: how does oak physiology vary with season, encroachment, treatment, so conifer removal, and climate? And then we wanted to not just look at one piece of the pie, but kind of the whole story about these ecosystems. And so we also took a stab at evaluating how woodland biodiversity varied with encroachment and treatment. And so for biodiversity, we looked at understory vegetation, mammals, and birds. And we were really interested in these treatments because there is widespread oak woodland decline due to conifer encroachment. There are kind of landscape scale oak woodland restoration efforts, and so we were interested in using this case study to kind of see how these woodlands were responding. And so, without further ado, because 15 minutes goes fast, I'll tell you what we found. So the first question, looking at oak physiology, this is looking at water potential. So on the vertical axis, we've got pre-dawn water potential, and pre-dawn water potential is the most hydrated status of plants in a 24-hour period. It's like the most flush with water that they'll be. And up near the top of the vertical axis is hydrated, and then lower down on the axis is water stressed. And so what we can see here, here's 2 years worth of data, so 2017 and 2018, and across both growing seasons in this Mediterranean climate, you can see this drawdown of kind of the plant available water. So we see decreasing water potential across our Oregon white oaks. So that's kind of the seasonal trend here. So winter rains come, and then across the dry, warm summer, those winter precipitation inputs are exhausted, and we see increasing plant water stress. If we look at this figure with our lens of encroachment levels and focus on the dark green columns for the heavily encroached stands, you can see that they actually have higher water status than the open or moderately encroached stands. So when we see widespread oak decline due to conifer encroachment, it begs the question, is this just due to a simple story of light limitation, like shading out the shade-intolerant oaks, or, especially in this age of increasing drought, severity and duration, and frequency, maybe is it that there's greater competition for water for these oaks, which is contributing to their decline under conifer encroachment. And so these data are showing that actually the conifers have sort of a buffering effect on oak water status. And again, we looked at many different metrics. We looked at live fuel moisture, VPD, like all sorts of things. I'm just showing you the tip of the iceberg here, but we see that the conifers actually had this sort of buffering effect on oak water status. However, it's not as simple as that, because then when we look at kind of how oaks are responding to climate, when we looked at vapor pressure deficit, so VPD, and you can kind of think of this as like how thirsty is the air, how drying is the air from a plant's perspective. There's a lot going on in this figure, but on the vertical axis, we have midday water potential. And so that's kind of in contrast to pre-dawn. That's like the most stressed out that a plant will be in a 24-hour period at midday. And then on the x-axis, we've got our pre-dawn water potentials. And then we've got that for 3 different stand types, heavily encroached, moderately encroached, and then open stands. And if we focus on sort of like the different color lines, so different vapor pressure deficits, the red lines, low VPD, means kind of wetter conditions, versus the blue lines, high VPD, being drier, more stressful conditions. And so what you'll notice is that in the open stands, all 3 of those colored lines are kind of all together. Like the oaks weren't very responsive to increases in VPD or drier, hotter conditions. But if we look at the heavily encroached stands, there's a pretty substantial drop between the red lines and then the blue lines. So we interpreted these data to mean that the oaks growing in the heavily encroached stands, while they had higher water status, they're much more sensitive to increases in VPD. So they might be set up for less resistance and resilience to increasing vapor pressure deficits, which kind of climate models project to be increasing with climate change. Okay, so that was the water status side of the story, looking at pre-dawn and midday. And then here's looking at stomatal conductance. And so stomatal conductance, this is of water vapor, but it's a gas exchange measure, and we can kind of use it as a proxy for productivity. And so what we can see here across 3 years of data is that as if you look at 2019 as the cleanest of the data sets, it was a little noisy in the first 2 years, but if you focus kind of on 2019, the most right set of columns, you can see that as after leaf emergence, stomatal conductance rates kind of vamp up until June, July, where they peak, and then from then on down towards October and leaf senescence, we see this tapering down of gas exchange, likely due to water potential, to limited water availability. If you recall back to the water potential data that I showed you, that kind of trend of decreasing water potentials across the growing season. So that's looking at the seasonal trends. And then if we look at this through the lens of what's going on with the different levels of encroachment, we again see that in the heavily encroached stands, so the dark green columns, we see, especially in 2019, really pronounced lower gas exchange values. So it's like, well, cool, the conifers kind of created this buffering effect for oak water status, but then the light limitation to the oaks due to the coniferous overstory shading them out really reduced their productivity. One thing that was really interesting that we found, and it definitely warrants kind of greater investigation, is that we found that there seemed to be some sort of physiological acclimation at this study site. So this is one study site, all the same species, and we found that the oaks growing in the heavily encroached stands were sort of physiologically marching to a different beat than the oaks in the open and the moderately encroached stands. So the far left panel and the middle panel, we've got midday water potential on the x-axis, and then we've got stomatal conductance on the vertical axis. And so oaks in the open and moderately encroached stands, they, as water potential increased, we saw stomatal conductance increase. And this is kind of an isohydric stomatal regulation strategy where you have plants regulating their stomata to keep water status at kind of a conservative level where they're not going to be risking kind of embolism and loss of hydraulic capacity. And then, so that's kind of, okay, cool, that's kind of how we would expect plants to behave. But then in the heavily encroached oaks, we saw kind of a really different trend, whereas water potential decreased, like became more stressed out, gas exchange actually increased. So these oaks were kind of behaving a lot more anisohydrically. Again, this is kind of the tip of the iceberg. We just saw this and we're like, huh, that's interesting. So it's a little bit encouraging in terms of thinking about resistance and resilience in this species to changing climate. So that's been kind of looking at seasonal trends and the effects of encroachment. And now let's talk a little bit about what happened when we went in and removed conifers. So we had 3 years of data, 2017, and then was our pretreatment data, and then after our 2017 measurements, they went in and thinned the conifers at about a 35% basal area reduction, so it was a pretty mellow treatment. And then we have 2 years of post-treatment data. And so let's take a peek at that. So based on our data, one thing, so here we're looking at pre-dawn water potential. So from what I've shown you so far, it might be concerning that, oh, wow, okay, the conifers seem to be buffering the oaks against water stress, and they also seem to be making the oaks more vulnerable or sensitive to increases in VPD. So that could be a dangerous combo if, as a land manager, you go in and take out a bunch of the conifers. Is that going to create a lot of increased water stress in your residual oaks? And so what we found, encouragingly, is that if you focus on the red and the green lines here, the dashed lines are the thinned treatments, and the solid lines are the control unthinned treatments. And so you can see that there was an ever so slight drop in pre-dawn water potential in the treated oaks compared to the non-treated oaks, but that is like a fraction of a megapascal. It doesn't seem biologically that meaningful. So at least at this site, with this level of basal area reduction treatment, we did not find that the oaks experienced an increase in water stress post-treatment in years 1 and 2. And then what happened with gas exchange? Again, I've already been shown my 5-minute warning. So I'll just kind of focus here on the heavily encroached stands. So on the vertical axis, we've got a ratio of post over pre gas exchange. So a value of 1 would mean no change. Anything under 1 would be a decrease in stomatal conductance, and anything over 1 would be an increase in stomatal conductance. And so we've got our 2 post-treatment years compared to 2017, our pre-treatment year. And you can see that for the heavily encroached thinned oaks, so the most far right pair of columns, that in year 1, there was an increase in gas exchange, and in year 2, they were able to at least maintain pre-treatment gas exchange levels. However, for the heavily encroached oaks that were not thinned, in both 2018 and 2019, they had significantly lower gas exchange compared to their thinned counterparts. So this is encouraging and cool information to have as far as land management goes, because with limited resources for oak woodland restoration, it can be challenging to decide where to direct those resources. And if there are heavily encroached stands, it's good to know, are they past the point of no return? Like, are they able to respond to that release and competition? And so these data suggest that, yeah, those heavily encroached oaks are able to kind of rally physiologically pretty immediately. That I need to mention too, though, that thinning those heavily encroached stands can be logistically and mechanistically really challenging because oftentimes the fir crowns are intertangled with the oak crowns. Okay, and then we also had a biodiversity component to this study. We used subplots to look at plant diversity. We used trail cameras to look at mammalian diversity. And then we used point count surveys to evaluate bird diversity. And I'll just kind of give you the highlights. The publication has tables of species encountered and species richness, species evenness, the Shannon-Weiner Diversity Index, all that kind of stuff. But we saw lots of cool animals, some highlights: bobcats, mountain lions, ring-tailed cats, bears. And it could be a product of a small sample size. It could be a product of the relatively light thinning treatment. But our biodiversity findings were a little bit underwhelming. So we found kind of predictably the lowest plant diversity in the heavily encroached stands. We found the lowest mammalian diversity in open stands. And then our bird diversity didn't really seem to vary with treatment or encroachment level. So there definitely could be improved investigations on this topic. And with that, my take-home points, this whirlwind of NorCal oak woodland encroachment and restoration, some things that maybe you can take away from this: conifer encroachment increases or reduces oak water stress and also gas exchange. Conifer removal does not meaningfully increase oak water stress and has the capacity to increase gas exchange even in heavily encroached stands. And we suggest after this study that it takes a lot to go in and restore these stands, taking out the conifers. And so we think that you'll get more bang for your buck if you're a little more heavy-handed than 35% basal area reduction. So that would be our recommendation. And with that, I think I have time for questions. Thank you for your attention. Thanks, Hugh.
Lucy, I want to know about you were talking about diversity. I need a microphone. I have the loudest voice in this room. Anyway, my question was about you have sort of diversity there. I'm guessing that was a relatively quick assessment, but my point is, I mean, you probably saw some pretty big compositional changes, right, across one of those states to another, I would assume.
We didn't actually see substantial differences in composition, and it was quick, it was a 3-year study, and in year 2, we got extra funding to do diversity, so we kind of tacked it on. So we didn't have pre-treatment data, unfortunately, which I wish we did have, but we just had the 2 years of post-treatment data, and we could compare control versus thinned, and then the different encroachment levels. But we didn't see that many that many differences in species composition, interestingly. And again, it could have been that, our sampling design was weak sauce, so at least we wrote it up in the paper and it can be used. When we were trying to put together our study design, there wasn't a lot to go on, so we were just kind of did the best we could to set up our investigation, and maybe other talks in this session will have better data than that. But this was our first stab, and I think we were limited by low power due to a small sample size, and we only had those 2 years of data, whereas I think that some of these biodiversity metrics might be slower to respond, especially to treatment effects. So if you had a longer arc of data evaluating responses of biodiversity, you might see more. Thanks for the question.
Two, but they're both quick. The 35% reduction in basal area is just among conifers. Is that correct?
Yeah, that was just dug fir removal. There were a couple incidental, oaks or hardwoods that were removed just by accident sometimes in the felling of the firs. But yeah the 35% was largely vast majority of that was Douglas fir.
And then you said you would recommend a higher percent basal reduction. Like, what would your recommended target be? 100%?
No, well, I think like at least like a 55% basal area reduction might be better. I think it would be more effective. This treatment, again, we're on timber company land, and so the treatment was to remove all Doug firs greater than 25 centimeters DBH because they were more interested in merchantable timber. So I think if you removed a lot of the younger Douglas fir too, that would be helpful.
That was a great talk, thank you. So I'm still trying to wrap my head around the fact that in the heavily encroached oaks, the relationship between stomatal conductance and water potential was flipped. Could you speculate about that?
Yeah, I know. There's been some head scratching about that. I think that those stands are so light limited that the only time where they are able to crank with photosynthesis or gas exchange is when it's like particularly hot and dry. It seems like, I think that the only time where they get that water stress during the midday is when it's hot and dry. And under those conditions, it's probably really sunny. Would be like make hay while the sun shines kind of thing. I think the only time when those stands are exposed to conditions where they can crank is when it's hotter and drier. But we were definitely surprised by that and want to do further investigation by that into those differences and trends. Yeah. I'm open, if anybody has any deep thoughts on that or insights, I'd love to hear them. Find me. Okay, well, thank you.